there! It looks like it's time for me to wake up. My name is Candy, and I live here by the Candy Sea. Today I'm here to talk about the protein orexin. Similar to the alarm clock, orexin plays an integral role in the body's sleep-wake cycle, which acts as an internal clock of sorts. Orexins are excitatory neuropeptides. In general, neuropeptides are large neurotransmitters that are synthesized in the cell body, packaged in vesicles, transported down the axon on microtubules, and finally deposited in the axon terminals. Like other neuropeptides, the functional orexin proteins are actually cleaved from a larger prohormone. HCRT, the gene that encodes for orexin, first produces a hypothalamic neuropeptide precursor protein, which is often referred to as pre-proorexin. Pre-proorexin is proteolytically cleaved into two orexin proteins, orexin A and orexin B. Orexin B is the smaller of the two, with a chain length of only 28 amino acids. Orexin A is slightly longer, with a chain length of 33 amino acids. The two chains share approximately 46% of the same sequence identity. Now, as good as orexins look in candy form, let's take a look at their true 3D structures. Orexin B is a linear peptide that forms two alpha helices, one slightly longer than the other. The C-terminal of orexin B is easily identifiable by the terminal sulfur-containing methionine residue. It is amidated here as a post-translational modification, so in the final product, this carbonyl group is converted into an amino group. Orexin A is slightly more exciting. Like orexin B, it also consists of two short alpha helices. In addition, orexin A contains 4 sulfur-containing cysteine residues that form 2 disulfide bridges. One bridge occurs between cysteine 12 and cysteine 6, and the other occurs between cysteine 14 and cysteine 7. Orexin A also undergoes post-translational modifications. It is amidated here at the C-terminal, and a proto-glutamyl cyclization occurs here at the N-terminal. Both of these modifications are common features of neuropeptides. Orexin-producing neurons are located exclusively in the hypothalamus, predominantly within the lateral hypothalamic area, or LHA. Humans have approximately 50 to 80,000 orexin neurons in the LHA. They receive input from a wide variety of brain regions that provide information about stress, circadian rhythms, and energy balance, among other things. Orexin neurons are able to integrate these diverse inputs and project widely throughout the brain to other areas. The most prominent projections are to the rostral reticular formation, where specific targets include the tuberomammary nucleus, raphe nuclei, locus ceruleus, and ventral tegumental area. Neurons in these areas all contain either one or both types of G-protein-coupled orexin receptors. The two types of orexin receptors are OXR1 and OXR2. OXR1 binds to orexin A with a much higher affinity than to orexin B, but OXR2 binds equally strongly to both orexin A and B. In humans, sleep-wake cycles are typically entrained to environmental cycles of day and night, or rather light and dark, through visual input. As the sun rises in the morning, special retinal ganglion cells are excited by light and synapse directly on the suprachiasmatic nucleus. The SCN is the master clock of the body that generates the circadian rhythm. During the day, when the SCN is excited by light, it inhibits the pineal gland, stopping it from releasing the sleep-promoting hormone melatonin. The excited SCN also inhibits the VLPO, another sleep-promoting region. With the VLPO inhibited by the SCN, the various nuclei of the rostral reticular formation become uninhibited. Orexin neurons in the LHA then begin to release orexin on their various targets in the rostral RF. Orexin excites these targets, and collectively this excitation is able to further inhibit the VLPO, repressing sleep even more. The neurotransmitters released by these nuclei, including dopamine, serotonin, and norepinephrine, travel to the thalamus and function to break the slow delta rhythms that are characteristic of deep sleep. With these rhythms broken, the fast rhythms of the cortex begin to increase, and the body is induced into a state of wakefulness and arousal. Like many physiological processes, the sleep-wake cycle has the potential to go awry. Narcolepsy is a disorder of alertness. It begins as a genetic predisposition, but its onset can be triggered during adolescence by other illnesses. It is characterized by the degradation of orexin neurons and the resulting depletion of orexin levels. The orexin levels of individuals with narcolepsy are 33% lower than normal, and those with narcolepsy and cataplexy have 92% less orexin. The most prominent symptom of narcolepsy is irresistible daytime sleep attacks caused by the intrusion of REM sleep into wakefulness. The most common treatments for narcolepsy include stimulants, antidepressants, and SSRIs to simultaneously stimulate alertness while also repressing REM sleep. Well, it looks like the sun is setting and I can feel the orexin dwindling away. Good night!